Welcome, and thank you for joining us for the fourth Agro Lunch and Learn webinar of 2023. We are delighted to have Eurofins Agri-Science Services as the generous sponsor of this webinar series. The Agro Division extends our heartfelt appreciation to Eurofins for their continuous support. My name is Solito Sumulong, the Director of Regulatory Services at AgriThority, based in Kansas City, Missouri. It is my privilege to serve as the moderator for today's webinar. So folks, please use the Q&A tool to submit your questions and comments and make the most out of your attendance today. Our team will actively monitor and respond during the presentation. Before introducing our speaker, let me emphasize the immense benefits of joining Agro. Enhance your science, expand your scientific network, and support your professional development. Visit agrodiv.org and click on Become a Member to join as an affiliate for only $20 per year, regardless of your membership status with the American Chemical Society. For the next fall meeting in Denver, we are planning exciting agro programming in line with the meeting theme, Elevating Chemistry. The organizing team is in the middle of planning sessions for this meeting. For any programming inquiries, please reach out to E.J. Gallows James Foster, the program chair for Denver 2024. Now, I am proud to introduce our speaker. Sidney Bluemink is a chemist and method developer at Smithers. He directs studies and develops methods for residue chemistry on soil, agricultural products, and animal tissues and fluids over a 20-year career that spans across tenures at Smithers, Covance, and Severn Trent Laboratories. He has a BS with honors in oceanography with chemistry from the University of Liverpool and happened to be a volunteer marshal for the 2014 Tour de France. So with no further ado, I'll give it to Sid Bluemink.
Thank you, Solito. So just sharing my screen here. And thank you, everybody, for joining me today. So today, we're going over navigating sample complexity in residue analysis, strategies and innovations for accurate and reliable results. So quick introduction and agenda. First, I'll just give you a quick slide about my employer, Smithers. I'll give you some background on residue chemistry, complex samples and the challenges they present, matrix effects and how we deal with them. We'll go over some sample preparation, extraction, and cleanup techniques, establish instrument analysis techniques. We'll go over some case studies. We'll go over some emerging trends and technologies relevant to residue chemistry, and then we'll conclude and wrap up with some questions. Right, so just quick slide about my employer, Smithers. We provide GLP-compliant residue chemistry studies for conventional and biological pesticide registration. We test a wide variety of crop protection products in plants and livestock. Recently, we acquired ResChem, based in Derby, the UK, and with that, we've expanded our global pesticide residue offering, and as well as the field cooperators network, so we can conduct more field trials. We have labs in the US and the UK, and we provide standard guideline and higher tiered environmental studies on a global scale. So thank you. If you're here, you probably know what residue chemistry is, but we'll go have a quick recap. The residue is what is left after an application of the compound of interest and after a withdrawal period. The residue is the analyte, is what we're interested in. And that's the context of this presentation. The material surrounding the residue is the matrix. And we're not interested in quantifying the matrix, but it's something we have to deal with when it comes to complex samples. So as far back as the 1950s, residue chemistry has been employed to quantify those chemicals in order to provide data for risk assessments and to monitor usage of those chemicals. And as we progress, we get stricter regulatory requirements, higher sample throughput, lower detection limits, and the increasing need for efficiency. It means we're employing ever more sophisticated and cost-saving methods and technologies. So when we talk about high complexity samples, what do we mean? I mean, the name's fairly self-explanatory, but I will go over some examples. Animal tissues, muscle, fat, liver, and kidneys, especially from common food animals, is something we often have to deal with. The components are proteins, phospholipids, fats, enzymes, and fibers. These can all present difficulties. Human and animal fluids, blood, plasma, waste products. Again, proteins, phospholipids, enzymes, inorganic components. We also have to analyze plant commodities, often for risk assessment and monitoring, pollinator studies, and again, proteins, phospholipids, the usual suspects. We also have to deal with dry commodities. Can also be problematic because with a dry, there's a high matrix load per sample weight. It means a lot of components that we have to deal with, such as cereals, spices, seeds. Soils and sediments, soils especially, very diverse. You can get a huge array of different types of soils that can present different types of problems. Again, monitoring, dissipation studies, mobility, adsorption studies. We have effluent and waste, sewage especially, again for monitoring and dissipation and mobility studies. And we also see a lot of bee diet and honey studies these days, mostly for risk assessment, monitoring and research. And with that, the problems they present is highly water-soluble carbohydrates, which can cause us problems. Other things we have to consider when it comes to sample complexity. This is not the main focus of the presentation today, but these are other things we have to consider, which can make an analysis more challenging or even less challenging. Some analytes can be more challenging than others for whatever reason because of their physical properties. We have to think about the limit of quantitation. A very low limit of quantitation can complicate a method, but in contrast, a high limit of quantitation can also simplify a method. So these are things we have to think about. Scarcity of matrix can make it problematic. Sometimes we have to deal with hazardous materials, difficult to handle samples, difficult to process, and we have to think about instrument performance and the amenability to our analytes. These are all things we have to consider when designing a method. So how do we address sample complexity when we're designing a method? Ideally, an analytical method needs to be able to quantify residues accurately and precisely, so we have confidence in our results, so we know they're true. A method should also be sufficiently rugged. It remains accurate and precise for different occasions, different people trying that method, and for different laboratories. It's robust so that minor errors don't adversely affect it. A selective method minimizes the probability of a false positive. In contrast, if you had a method that is not selective, what happens is you're in danger of mistaking a matrix component as your analyte, for instance. And you want your method to be sensitive, that minimizes the probability of a false negative. If your method is not sensitive enough, then the analyte might be present in your sample, but you just can't detect it. And complex samples challenge a method's ability to have these attributes. Ultimately, it's about getting that signal to noise, signal being the analyte, the noise being everything else that we're not interested in. One of the most important aspects in analysis is matrix effects. There's the IUPAC definition. Matrix effects are the combined effect of all components of the sample other than the analyte on the measurement of the quantity. So they introduce bias, they influence the method performance. And these can be initial extraction and sample workup challenges. The most important one we usually have to deal with is the endpoint, is the instrumental chromatographic matrix effects from our final extracts. And we should also consider that reagents and materials can also be a source of matrix effects. So first thing we address is the extraction challenges. We have to think about extraction efficiency. So efficiency being our ability to separate our analyte from the matrix. And this can be affected by choice of solvent, temperature, agitation method, and sample comminution, that is particle size. The correct extraction regime is vital, because if you get an incomplete extraction, this will lead to an underestimation of your levels present in the matrix. ideally, the extraction regime should have been demonstrated on a radiolabeled incurred samples. And this is especially a requirement for compounds undergoing risk assessment at the moment. Myself, I work mostly in the risk assessment field, and this is something we very much have to think about. We also have to consider that if the extraction regime is too harsh, unwanted matrix components can be carried through, requiring more cleanup. There's also challenges when we are working up samples. This is after the extraction, and if we have high matrix load samples, we have to think about suppression of reactions, if we have to do a derivatization or a deconjugation step. We'll go over this in a bit more detail later. If you're doing solid phase extraction, you might just get overload or overload of your filters. If you're doing liquid-liquid extraction, you may get emulsion effects. We may get suspensions in final extracts. These can clog up your columns. We have to think about absorption of analytes onto container surfaces and saponification. If we're dissolving tissues and fats with alkaline metals, we get formation of soap, which causes problems. So instrument matrix effects, this is the end point, and this is where we can really know what's going on with our samples. So when developing methods, complete elimination of instrument matrix effects is difficult to achieve, if not impossible, and to aim for that is often not economical or practical. Sometimes we work on a fit for purpose approach that it's often not practical to over optimize a method. That being said, we should always aim for a sufficiently clean sample, because if they're not sufficiently clean, we might have to worry about incurred samples where from different sources, they may be prone to an unknown or unpredictable matrix effects. This can actually affect method ruggedness and robustness in the long term. And we have to think about that if our samples are not very clean, this can affect instrument operating costs, which can be considerable. More matrix load means we use more consumables, more instrument downtime, maintenance costs, and operator time all goes up. And it's a balancing act. So what are matrix effects? What do we commonly see with them? So in something like liquid chromatography and mass spectrometry, we see signal suppression. This is competition for ionization, and we also see sometimes signal enhancement, matrix-assisted increase in ionization efficiency. In gas chromatography, we often see enhancement with matrix effects, where the matrix actually acts like an analyte protectant and protects it from thermal stress. And this actually gives an improved peak shake and signal to noise. Suppression is something we don't see as often in gas chromatography. And we also have to think about general chromatographic effects. Isobaric co-eluting components in mass spectrometry. Isobaric means that they have similar mass characteristics, and if you don't have sufficiently high resolution of your mass spec, the instrument could mistake a component for your analyte. And an example there in the chromatogram at the top, we've actually isolated the analyte from an isobaric peak interference using chromatography. And we also have to think when we're using less selective detectors such as LC ultraviolet or flame ionization, co-eluting peaks can just interfere because these detectors are less selective. And we also have to think about overload of, if your sample is too dirty, you can overload the column, the detector, or your GC liner. And you can see an example there in the chromatogram where we have some matrix overload. So we spoke about extractability. In risk assessment where I work, as I said, we have to think about this. So how do we look at extractability? How do we assess it? As I said, ideally the extraction regime has been demonstrated to give acceptable extraction efficiency, often with radiolabeled analytes in incurred samples, because what we can do is we can take the total radioactive residue by combustion and we actually compare it against a usually extensive extraction regime. And this gives us all of the radioactivity that was applied to a system. But we also have to think about that the total radioactive residue may not represent the residue definition for dietary risk assessments. These are the actual residues we're interested in a risk assessment or indeed in a monitoring method. And what we can do, an extraction like that, which is too extensive, is not very efficient. So we can actually look to do a cross-validation where we compare the extensive extraction regime with a simplified one. And the end result is that we have a method that's more suitable for high sample throughput. And this is something we have to think about in risk assessment. So we can also assess and measure matrix effects on an instrument, and there's different ways we can do that. Post-extraction spike is something which can work for almost any instrument, where we compare a non-matrix standard with a standard of the analyte in matrix, and from that we can simply calculate percentage enhancement suppression. And we can use this to compare various matrix sources. If we see a variable effect from different sources, this can maybe tell us that we need a better cleanup. We can also do post-column infusion. So what we do is we inject a blank matrix onto the instrument and the column, but at the same time we're infusing a constant amount of our analyte. And what we can do is we can monitor the response of the baseline, and it can inform us of where matrix components are eluting and how they're affecting the analyte signal. We can also do a phospholipid scan, similar to post-column infusion. We can simply qualitatively scan various transitions associated with phospholipids, and we can see where they're coming off. In biological samples, this can be especially effective because phospholipids can produce a lot of problems. And as you see on the chromatogram on the right, that's a phospholipid scan, and you can see a rise in baseline, and that's showing us that, yeah, there's a problem there. We've got lots of phospholipids coming off near the end of the run. We can also simply do a full scan of the blank matrix using a mass spec, and again, we can identify those matrix components, and we can test our method just using various sources of our samples. This can be important, and indeed is a requirement for lots of validations. You have to test the various sources. Reason being is if you're dealing with a plasma sample, even a plasma sample coming from myself can differ depending on the time of day, what I've had to eat, if I'm tired or if I'm ill, in field trials, we can perform field spikes of actual controls, and this can have the same effect. So mitigating matrix effects, how do we deal with them? The aim, as I said before, is to deliver a sample to the instrument that's clean enough to meet the targets of those method attributes that I talked about: accuracy, precision, ruggedness, robustness, selectivity, and sensitivity. As well as we try to reduce the matrix effects, but there are also ways to compensate for them. And it's also important to point out that advances in instrument performance can help reduce sample workup time, and this is good for operator time. We let technology do the work if we can. Even so, as I said before, it's still important to deliver a clean sample, but we can make the analysis more simplified, make it more cost effective, and it's often easier to automate. And we have a variety of tools available to us now to solve most analytical problems, and we'll go over them now. So one of the first techniques available to us is matrix matching. We designed the method so that the calibration standards contain matrix components from a blank sample, and this way it compensates for matrix effects, standards versus your actual samples. So if the standards and samples, if they undergo the same effects, we cancel bias out. Again, this method should not be a substitute for an effective cleanup, since effects can vary across different sources. And this is as opposed to using calibration using pure solvent non-matrix standards. In risk assessment, especially, regulators usually require matrix matching unless we can demonstrate it is not significant. Typically, this is a post-extraction spike of known amounts of the analyte into control matrix. I should be pointed out, in gas chromatography, as well as matrix matching, we can just use an analyte protectant that will actually make the effects more uniform across all your samples. There are drawbacks. This can be labor intensive, and it's also problematic if our matrix is scarce, but it's a tool. It's one of those tools available to us. Another tool that's available to us, not all the time, but can be helpful, is using an internal standard. So what we, excuse me, what we do is samples are spiked with known amounts of a different substance, and we measure the ratio of the analyte and the internal standard. A crude example would be is if you take a liter of water and you add an amount of your analyte and an amount of the IS. It doesn't matter if you then spill half of it. The ratio between those two are then the same. And this is how an IS works. It compensates for changes to your analyte, corrects systematic and random error from sample preparation and instrument analysis. So the first type of IS we can use is a structurally similar or even dissimilar analog IS. These are useful as injection markers, and it's usually as a post-extraction spike, but they don't necessarily compensate for instrument matrix effects, and typically they don't co-elute in the chromatography. They can, in special cases, compensate for matrix effects, but we have to be very careful about our selection. They don't readily compensate for MS ionization effects because they don't co-elute. And for environmental samples, the IS should not be a compound that can be readily found in the environment for obvious reasons. And we can use dissimilar analogs as well, but generally they can only be used as injection markers. And an example is caffeine with an internal standard using adenine, as they have some similarities. So the other type of internal standard we use tends to be more powerful is isotopically labeled IS. Structurally identical to your analyte, but has different isotopic components. Most commonly, the hydrogens in your molecule have been replaced by deuterium. An example here on the right is anisole, where the hydrogens have been replaced with deuterium and they have a different mass. So chemical properties virtually identical, but they have a different molecular mass. These type of IS will not exist in the environment, and very often they will compensate for matrix effects because they co-elute and they undergo the same ionization process. Yes, and this is indeed an invaluable tool for mass spectrometry, and indeed it will only work for mass spectrometry. There are some potential drawbacks. Even used in conjunction with matrix matching, it should not be a substitute for an effective cleanup. It should not be used to compensate for a very poor method precision, because we should be using an IS to improve a method. It shouldn't be there as a sticky plaster. And in some cases, an IS can interfere through mass spec crosstalk. This is something we see if we have a diverse isotope distribution in our molecules, such as they have chlorine or bromine. These can cause problems. We should consider, though, that they're expensive and not always readily available. They won't always be there for every analyte. Indeed, if we're working on a multi-residue method where you might have to deal with 200 analytes in a single run, it's very unlikely that every single analyte is going to have an analog isotopically labeled internal standard. And even if it was, that would be very expensive and costly to do. So it's certainly not a panacea, but it's still a very valuable tool is using an isotopically labeled IS. And with that, we'll just go on to a quick case study. If you work long enough in this industry, you're probably familiar with cypermethrin. And in this case study, we had a requirement to validate a method for the analysis of cypermethrin in water with a limited quantitation of 0.01 parts per billion. Very, very challenging. Cypermethrin is a synthetic pyrethroid widely used as an insecticide, and it's a challenging compound to analyze. Anybody who's worked with this will know that, and has been studied in many matrices. Indeed, I've been working with it for 20 years plus, and it never seems to go away. It exists as 8 isomers and will manifest as 4 peaks in gas chromatography, and you can see there what it looks like in a typical chromatogram. I should point out, though, I'm aware water is not a especially complex matrix, but the combination of the compound and the limit of quantitation certainly makes it challenging. So yeah, the target molecule was very challenging for detection and precision. In 2012, an isotopically labeled internal standard of cypermethrin was released to the market called trans-cypermethrin D6. Indeed, there is I believe there's another presentation on the Agro website which talks about using that particular IS. And we found this compensates for matrix effects, analyte losses, and instrument precision. It came in very useful when it first came out. Nature of cypermetry means it's difficult to achieve the acquired sensitivity. So we employed programmable temperature vaporization, injection with chemical ionization, MS. These are components of a GC, which can come in very useful sometimes for this type of targeted analysis. With cypermethrin, cross-contamination is a frequent problem with cypermethrin. So the solution was only used fresh and disposable glassware. We also have to consider that cypermethrin tends to adhere to container surfaces in aqueous environments. So it's a challenging compound to work with. The workflow was 40 mils of water. We extracted it from a 50 mil Falcon tube containing already 10 mils of hexane to prevent adherence to container surfaces. We added the IS. We did a very simple liquid extraction and a concentrate evaporation and a reconstitution, and we got really good precision and accuracy, excellent linearity, and we found the internal standard was able to compensate for systematic and random error, cancel that matrix effects, and these effects were even indeed tested on various sources, and we eliminated cross-contamination. This is an example where use of an internal standard came in very handy for us and really helped us with a very challenging limited quantitation. So one of the other tools available to us, and especially in multi-residue methods and monitoring, is catchers. This was cheap, quick, easy, cheap, effective, rugged, and safe. Was first described in 2003. It was a simplified and robust method used for MRMs, and it took advantage of advances in instrument performance. Relatively easy to automate, and it indeed was originally designed for MRMs, but may not be targeted to a particular analyte, so may require more optimization and manipulation, depending on if we have more targeted analysis, say in risk assessment. But it can still be used as a standardized approach for that targeted analysis. We have to consider, though, that the use of the simplified extraction regime in catchers could be in conflict with the need for extraction efficiency. As I said before, we have to think about extractability, and in metabolism studies, we may require more extensive extraction. So then we may need to make adaptations. Mostly unsuitable for hydrophobic analytes in fatty, oily matrices and many hydrophilic analytes, but this is addressed in later iterations, which we will go over. Typical workflow for catchers: 10 grams of sample, agitate with 10 mils of acetonitrile. We do a salting out where we add salt, various combinations of salts, and this can separate the water from our extraction solvent. We can spin this out. We can apply a cleanup using dispersive SPE, and the extract can go for LC or GC analysis. And that's just an example. We can use various solvents, different salt combinations. We can do pH control. So there are very many iterations of this. This is very much an example. There have been further developments of catchers recently. One of them is catcher-zer, or mega method is probably a better name. So this accommodates an even wider range of polarities and analytes, including pharmaceuticals now, and it takes advantage of even more recent advances in performance, as the original catchers is already 20 years old. So this takes advantage of, there have been more advances since then. This generally, we increase the solvent volume to the sample weight, and this aids in extraction efficiency. It's also recommended that we increase sample comminution using liquid nitrogen for ultra cold processing. This makes our samples more homogeneous, more representative, because we're dealing with a smaller amount of sample. It decreases particle size, increases surface area, and increases our extraction efficiency. And the initial salting out step is actually skipped for polar analytes, because otherwise we would lose them. But we do use a salting out step for a GC analysis, which works for non-polar. And here is a typical workflow that we would see for this new catchers iteration. As you can see, there's this is very much an example of where the extraction is even more simplified, but it takes advantage of the more advances in instrument performance. Right, and there are more iterations of catchers that I'll go over. One of them is quick oil, Q oil. This works well for oily samples such as rapeseed, sesame seeds, poppy seeds, nuts, mostly with hydrophobic analytes. Especially relevant for the analysis of antibacterial fumigants such as ethylene oxide, which we have to monitor due to carcinogenicity. And there is another example workflow of how we would use Q oil. And again, we take advantage of advances in instrument performance. And finally, with catchers, the other iteration is quick polar pesticides. For very hydrophilic analytes that even the mega method struggles to cover, these are analytes that can include the paraquat, diquat, and perchlorates. Very problematic to analyze by conventional LC, so will often require ion chromatography or Hillick mass spectrometry. And there's even a step where matrix components such as lipids, sugars, and proteins are eliminated by a very old-fashioned freezing out step, but nevertheless effective. And again, there's an example workflow there of how that works. These are all different iterations of catchers. So that brings us on to another case study. This is just to highlight how powerful catchers can be when used in the right combination. So a requirement was to validate a harmonized method for the analysis of common herbicide in bovine fat, muscle, liver, and kidney. The extraction regime required by the regulators was known, and we found it could be adapted to be compatible with standard catchers workflow. And you can see the workflow here. 20 mils of solvent was initially used to extract from the tissues. This was aided by the use of steel balls and vigorous shaking using a Geno grinder. We centrifuged it, and the supernatant was then subjected to the catcher salting out, and then we were able to take an aliquot and clean it up, and we were actually able to further dilute the sample because the instrument performance was exceptional. And the result was that for a start, we couldn't find a suitable IS. But this didn't adversely affect the results. There were no matrix effects because we were able to extensively dilute the sample because the instrument performance was that good. We had really good precision and accuracy and linearity, and even the lowest calibration standards showed a signal to noise of 50:1, as you can see there on the right. And the standardized approach that we used easily accommodates higher throughput. So that's an example of where we use catchers very successfully. So other tools that are available to us: solid phase extraction, been around for a very long time, still very powerful technique where we draw liquid extract through a cartridge containing a salted sorbent. The sorbent type is selected based on the affinity of the analyte it has for the sorbent and the solvent. This way we can eliminate unwanted matrix components. We can retain the analyte, we can actually keep the analyte, the matrix, onto the sorbent while we elute the analyte. The various orthogonal SPE designs we can use to remove matrix. By orthogonal, I mean we can use different separation mechanisms. We can take advantage of the different physical properties of an analyte. We can do things like exchange solvents and concentrate the analyte. It's very powerful for targeted analysis, but indeed also used in multi-residue methods, but perhaps not as versatile as catchers. But while it's not as versatile, we can still analyze a wide variety of analytes, perhaps more in terms of compound class. And there's definitely more modern approaches available using new sorbents. We can use mixed sorbents, well played, and plenty of automations available now. Another tool available to us when we're having problems is standard addition method. So in this, a series of known incremental amounts of our analytes are actually added to multiple replicates of an incurred sample. This is similar to constructing a matrix match calibration line that we discussed earlier, but the matrix already contains an unknown amount of our analyte, and the unknown is what we want to calculate. The real concentration of the analyte of the incurred sample can actually be mathematically extrapolated. We use this when matrix effects are proving very variable due to different sources. When there's an IS is not available. Typically, it's for analytes that are very hydrophilic and chromatographic and detection selectivity is poor. We also use it when the analyte actually exists endogenously in your sample type. In other words, it's not possible to obtain a true control blank matrix. Excuse me. Indeed, if you took a plasma sample from me, you'd be hard pressed to find a blank plasma sample of different hormones. So that's an example where standard addition methods are very useful. There are drawbacks, very labor intensive, and requires multiple replicates of each incurred sample. So if your sample is scarce, then you're running into problems. And here's just a quick graph of how we would, a quick illustration how we would calculate concentration of a sample based on the standard addition method. You have your endogenous analyte signal, and then you add your incremental amounts of your exogenous analytes. You construct that line, and then you find the x-intercept, and that gives you an extrapolated concentration of the analyte. So it's a very powerful technique if you're running into a lot of problems. Also, other sample techniques, sample workup techniques, quite mundane, but still important. Things we have to think about when dealing with complex samples. Dilution, very simple technique. Sometimes if the instrument performance is that good, that's all we need to do with a sample. We can actually just dilute out the matrix effects. And that's very useful if we have exceptional instrument performance. Filtration, eliminate undissolved matrix components. We have protein precipitation steps, which we use in biological samples where we denature proteins, and we can actually have centrifuge precipitate them out. Evaporation steps where rotary evaporation or just nitrogen stream dry blocks or turbovaps. We can concentrate our analyte, or we can even swap solvents if we need to. And of course, old-fashioned liquid-liquid partition where we mix immiscible solvents, where we want to maybe transfer the analyte from one solvent to another. Also, we have to think about reactions. Sometimes we have to derivatize our sample, where we transform it to make it more amenable to instrument analysis. We increase the molecular mass, we maybe make it more stable, increase the volatility, we get more MS fragment ions and just better detection. But we do have to think about matrix interference, selectivity and yield of that reaction. And a typical example here is glycine and its derivative, which makes it more amenable to GC analysis. We also have enzymatic hydrolysis or deconjugation, and this reverses biological glucuronidation. So a typical example is with codeine. If I were to ingest some codeine and somebody would take a plasma sample, they would find the codeine glucuronide, and then so we do a deconjugation to get back the free codeine, so we can analyze it. That's an example, but this can indeed be used for plants and all sorts of biological samples. So another tool available to us is increasingly these days is hybrid mass spectrometry. So what we do here is we combine two or more mass separation techniques in one instrument. Traditionally, we've had triple quadrupoles, but we can now combine that with ion traps, orbit traps, and time-of-flight mass spectrometers. They're usually high mass resolution. They improve that selectivity. They were once the preserve of metabolite ID, but now regularly used for sample quantitation, especially in multi-residue methods. Their high scan rate is especially powerful when you're dealing with hundreds of analytes in one go. And that can be coupled with a catcher's analysis technique, make it very powerful. And the diagram shows a typical layout that you might see. This one's for an eclipse, and it has various mass analyzers. And that brings us on to our last case study, where we had a requirement to analyze soil for earthworm testing of a significant fungicide metabolite. This proved to be very challenging to separate from matrix using traditional triple quadrupole. Triple quadrupole uses one AMU, one unit resolution. And in this case, we were getting too many isobaric interferences, components that had a similar mass. So we employed a high resolution quadrupole time-of-flight hybrid, and this improved our selectivity. So you can see on the two diagrams there, the two mass spectra. The one on the left is the compound scanned using unit resolution, and on the right you can see the same compounds scanned using high resolution, and the horizontal axis is has the same scale. So you can imagine this sort of wide spectrum here. This means that there are other components that could have similar mass characteristics, and indeed the unit resolution instrument would mistake other components for that analyte. But if you look on the right with high resolution, it's very unlikely, extremely unlikely indeed, that any other components would be mistaken for that analyte. And we solved these problems. We did a simple extraction and dilution. We had very good precision and accuracy, and indeed we could see the lowest concentration matrix standard at 0.5 parts per billion. And here's a chromatogram of that standard. Again, trying to illustrate, on the top, you can see that standard where we're using a unit resolution. And on the bottom, we've applied mass filters, and you can suddenly see how much it's cleaned up our sample and how we can see that peak much more clearly. And that's really a powerful example of how high resolution hybrid technology can offer a lot of advantages. We should also talk about established instrument techniques, high performance, ultra high performance liquid chromatography, high pressure pumps. This really allows for greater resolution in chromatography, really powerful. Last few years, with traditional triple quadrupoles, we see the designs often go toward lead towards increased ion transmission. So they use larger ion inlets, they use a stronger vacuum system, also improved ion optics and electronics means that we even triple quadrupoles can continue to advance. We also have ion mobility available to us. This is a module that separates ions based on their mobility in a buffer gas. They can be combined with quadrupoles to give that extra layer of selectivity. They're useful to separate stereoisomers. They can eliminate interferences and difficult to retain analytes, such as those I've mentioned in the quick polar pesticides method. We also have available to us two-dimensional chromatography, known as often peak parking. We can isolate peaks into sample loops and then switch them and divert them to another column to give an orthogonal analysis where we switch our separation techniques. And again, it's very powerful. I should point out, for two-dimensional chromatography, this is very useful for a risk assessment. Might not be so useful for multi-residue methods where you're dealing with hundreds of analytes. So it really depends. Some instruments are more useful for some aspects and not so useful for others. We have gas chromatography, still very powerful when we couple it with different sampling techniques, and it can easily accommodate catchers and their iterations. We also have some emerging trends and technologies that we're speaking about in the residue field. We have a diethylamine bonded phase LC column. This improves retention and selectivity of polar analytes, which have traditionally always caused problems. These don't require mobile phase additives, which can suppress signals, and this is proving especially useful, again, for quick polar pesticides types analysis. We also have microflow and nanospray and matrix-assisted laser desorption. These techniques are targeted more towards macromolecules and proteomics, which we don't see much in residue chemistry, but we may see that more in the future, especially with the increased use of biologics as medicines. So, these techniques have been around for a while, but then generally not used much in residue, but we may see that more in the future. And also supercritical fluid chromatography, where we use liquid CO2 as a major mobile phase component. This can offer unique selectivity, improve mobile phase desorption, and again, stereoisomer separation. Finally, some other trends. So for gas chromatography, we have mid-column backflush and low pressure GC. Technologically, not especially advanced, but they're just different new techniques, new ways to handle samples. And these can also prove especially useful for multi-residue methods in GC because they can allow for faster analysis. One I'm particularly excited about, which is gaining traction, is gas chromatography, atmospheric pressure chemical ionization, where you couple a GC with an APCI source. which you usually see in LC applications. And this is a softer ionization technique, which improves selectivity. And indeed, it can be combined with catchers and megamethod and the quick oil. And we also have to think about automated sample preparation, pipette robots, auto samplers, SP stations. These have been around for a while, but they increasingly say for risk assessment methods where we didn't always have routine methods, it would prove difficult to adapt automation to non-routine methods. But it's definitely something that's already widely available for multi-residue methods or monitoring methods. So I'll conclude now. Sample complexity challenges, methods' ability to produce reliable results. We now have a wide variety of tools available to us to solve most of those problems. And with the right combination of experience, knowledge, mindset, support, technology, and instrumentation, we can address most sample complexity problems. And in future, implementation of new trends in technology will add to the tools already available. So yeah, thank you very much for your attention. Just point you to our website, smithers.com, where there's lots of other resources available. And indeed, you can contact myself if you want, if you've got any extra questions, apart from what we will ask now. So yeah, I'm happy to take questions. So thank you very much for your attention.